Dementia is an insidious disease that gradually and subtly steals a lifetime of memories, our personalities, our ability to live independently, and eventually our lives. It describes a set of symptoms such as forgetfulness, confusion, and disorientation, which occur when the brain cells are damaged by disease or a series of strokes. Around 47 million people are affected worldwide, a figure that's expected to double every 20 years, with much of the increase occurring in developing countries. It costs $818 billion worldwide, 1.1% of world GDP. Put simply, dementia is the biggest medical and social challenge facing humankind. By studying the basic building blocks of life, mini brains grown in a lab, and worldwide populations, researchers at Cambridge have one aim: to stop dementia.
Dementia is actually a global name that we use to describe and group together a series of different illnesses that all affect the brain in the same general way.
The most common form of dementia is Alzheimer's disease. About 2/3 of dementia is caused by Alzheimer's disease. Then there are other forms as well. Vascular dementia is probably the second most common cause. And then there are rarer ones as well, such as frontotemporal dementia, dementia with Lewy bodies.
Dementia is not the same as normal aging and is considered a distinct condition. However, aging itself is a major risk factor for dementia. The change in risk is not simply a change in chronological age. There's something that is occurring when one ages that dramatically influences one's likelihood of getting dementia.
But it is not only the old that are affected. Around 1% of cases of Alzheimer's disease are caused by a genetic variant found in family lines. Andrew King is one of these people. His wife Sarah started noticing some unusual behaviour: car accidents, confusion, and underperforming in his role as a geneticist at the Sanger Institute where he worked. After a long series of tests, he was eventually diagnosed with early onset dementia. He was just 43 years old.
Dementia didn't even cross the horizon because we thought it was kind of, if anyone had asked, we thought, oh, that's an old person's illness. Got the diagnosis, and then within a couple of years he couldn't walk properly, he was doubly incontinent, having problems eating. I went into the care home within, so he's been about 8 years ago, so he must have been 45. He was having to have a fed by tube, and then he's been pretty much the same since then.
Although much is known about the mechanisms behind the development of dementia, there is no known cure.
It's very difficult to think of another example in the history of medicine where we've got to the level of understanding about the disease that we now have for Alzheimer's, and that hasn't led to therapeutic benefits.
The traditional approach to dementia is nothing can be done. But today, you are telling us, you are turning around this approach to say something can be done.
At the 2013 G8 Dementia Summit, health leaders from across the world pledged a research ambition for a disease-modifying therapy for dementia by 2025. Alzheimer's Research UK has formed a new drug discovery alliance that will make a major contribution to delivering this ambition, a UK-wide network of institutes dedicated to early-stage drug discovery.
So our Cambridge Drug Discovery Institute is part of an alliance of 3 of these DDIs with Cambridge, Oxford and UCL. And really what we're doing is building and embedding drug discovery expertise and experience to work alongside the great researchers here who are finding out more about the mechanisms of disease.
The purpose of the institutes is firstly that they work together, they cooperate to some extent, and so it's to bridge the gap which currently exists between academic scientists and the initiation of drug discovery projects.
The initial stage in drug discovery is to understand what actually happens in the brain to cause the disease. In 1906, Dr. Alois Alzheimer first discovered what we now know to be the hallmark Alzheimer's proteins. He was assigned to the case of Auguste Dieter, a 51-year-old woman who was admitted to the Frankfurt Mental Institution with typical symptoms of what we now call dementia. Upon her death, Alzheimer discovered strange plaques and tangles in her brain. Plaques are abnormal clusters of the protein beta amyloid that build up between nerve cells in the brain. Tangles are caused when fibers of tau proteins twist together, a process first identified by Dr. Michel Goder at the MRC Laboratory of Molecular Biology.
In the 1980s, we showed that the tau protein is an integral component of these filaments that form inside nerve cells, from where they spread to distant brain regions. And it is this spread of brain to distant brain regions that causes the symptoms of Alzheimer's disease. So understanding the mechanisms underlying the spread might allow one in the end to prevent the spread from occurring and therefore the symptoms of Alzheimer's disease from appearing.
Even in a healthy brain, plaques and tangles develop normally with age, but they are found in much greater abundance in people with Alzheimer's. They start accumulating in areas of the brain associated with memory and learning, and then spread to other areas. In the Department of Clinical Neurosciences, Professor Peter St. George Hislop looks at how small changes in the instructions carried by some genes can lead to the creation of these abnormal proteins with abnormal functions.
Over the last several years, we've described and discovered about 20 different genes that are associated with risk for Alzheimer's disease. And we're doing the same thing for the other causes of dementia, like frontotemporal dementia. And so the next step is now to take those genes and ask how it is that variants in those genes increase the risk for developing the disease. We know for some of the genes that they alter the processing of a protein called amyloid precursor protein, or APP, which generates this neurotoxic amyloid beta peptide. But some of these other genes, they seem to have a different way of acting. And so we're going to try and work out what that mechanism is, how they work. And the longer term is, once we understand the function of each of those genes, to then be able to try and find a way in which you can block that abnormal function. And ultimately, at the end of the day, what we might be able to do is to say somebody has a variant in this gene, they're at slightly increased risk of developing disease, so that person should be given that treatment, and another person who has perhaps another genetic variant that increases their risk might require a different treatment that is specific for their particular type of the disease.
Professor Chris Dobson and his colleagues Dr. Thomas Knowles and Professor Michele Vendriscolo at the Centre for Misfolding Diseases have been studying amyloid and similar proteins to understand how they fold into functional structures and what causes some proteins to misfold into the wrong shape and generate disease.
When proteins are made, they're made as long chains of amino acids from the information contained in our DNA. And to function, they have to fold up into specific structures. We have these natural protective mechanisms that keep our proteins in good shape and functional, and if they misfold and aggregate or clump together, then they're cleared. But at some point, these can become overwhelmed, and then you start to accumulate these species in the brain and in dementia. And even worse, they're sort of catalytic, and the disease progresses very rapidly. Once we started to understand the, if you like, the mechanism by which proteins misfolded, then we began to think about how that can be prevented. And what we've been doing then is actually to try to find small molecules in particular that can inhibit specific aspects of the misfolding reaction. And there are two particular aspects of this that we're very excited about at the moment. One is the idea that you can take a molecule a bit like a statin, it's sometimes called a neurostatin, or we've called it neurostatin, that reduces the risk that you would actually get Alzheimer's disease. And the other is that if one clearly has symptoms of the disease, can we develop small molecules that will reduce the rate at which it progresses, or even how we could actually enable the brain to recover from these processes. And we have a number of these molecules that we've found inhibit the processes in the test tube. We've been looking at how they can do that in simple model organisms such as fruit flies or the nematode worm. And we've been very excited about the results because we seem to be able to suppress these processes very effectively. And so the next step is to take those further and to see how we can develop them potentially as drugs to use in the clinic.
The cells in our body have their own natural way to remove dysfunctional proteins before they become toxic and start to destroy brain cells. This process is called autophagy and is a key stage in treatment development that is being researched by Professor David Rubenstein's team.
Autophagy is a process that cells use to get rid of parts of their cytoplasm. That's the bit of the cell that's outside the nucleus. And in doing so, the cells form a double membrane structure which engulfs a portion of the cytoplasm to form a vesicle, which we call an autophagosome. And then it delivers the autophagosome to the lysosome, which is the cellular incinerator for degradation. And this allows the removal of parts of the cytoplasm, and in this case of the neurodegenerative diseases, these aggregate-prone toxic proteins. We've worked very hard to understand the basic biology of the process, to identify therapeutic targets, but also to try to identify drugs that you might be able to use in people that can enhance this process, reduce the levels of these toxic proteins, and ameliorate the signs or symptoms of the disease.
It is thought that the clusters of abnormal proteins that cause plaques block signals passing between nerve cells in the brain and promote a process that leads to disintegration of these cells. The twisted tangles of tau proteins are thought to block the movement of nutrients around these brain cells, which then die. Professor Giovanna Malucci and her team are looking at slowing or preventing the process of cell death and whether damaged cells can be repaired.
Well, there are two main strands to the laboratory. The first has identified a really fundamental process that happens probably in almost all of the diseases that cause dementia or neurodegenerative processes. They're called protein misfolding disorders, and how that misfolding of these proteins causes the brain cells to die. We've also discovered how to manipulate that process, both using genetic tweaks, but also using chemicals or drug-like compounds, and we've cured degenerative diseases in mice. But this compound is toxic to the pancreas. It protects the brain, but it's toxic to the pancreas. We've moved on from that. We found another compound that protects both brain and pancreas, but it's so insoluble. So the question now is drug discovery and improving the kind of compounds that will do the same job, but with minimal side effects.
Once potential compounds or drugs are identified, the next step is to test them to see which are best to take forward into clinical trials. It is hoped that research into each of these mechanisms and processes that lead to dementia will ultimately reveal drug candidates that can be tested on humans in clinical trials. But this takes time and money. Within the Gurdon Institute, Dr. Rick Livesey's research has identified a remarkable modelling technique using stem cells to replay Alzheimer's disease in the laboratory.
What we do here is we make human neurons in the lab, but we do that starting from individual patient skin cells. And so based on technologies developed around the Nobel Prize from Professor John Gurdon, the founder here, we can turn skin cells back into stem cells. Now, stem cells, in principle, can make every cell type in your body. In our case, we use them to make the part of the brain that's affected by Alzheimer's disease. The overwhelming majority of Alzheimer's disease affects people in their mid to late 60s, but there's a rare inherited form of Alzheimer's disease which affects about 1% of people with Alzheimer's. So this is the familial or the genetic form of the disease, which runs in families, and it's due to changes in just one gene within your DNA. What that means is people with these familial Alzheimer's disease mutations develop early onset Alzheimer's disease, typically in their late 30s or early 40s. So what we do is we make stem cells from individuals who carry this familial Alzheimer's disease mutations. what that allows us to do then is to compare the disease to what actually happens within the 99% of people who have the sporadic or late onset form of the disease. The advantage of using stem cell systems to study Alzheimer's disease means that we're actually studying real human neurons within the lab, so the neurons that are actually affected by the disease. We can also study it over a much shorter time frame, so typically months in our case, say, rather than years. Stem cell systems are also useful in accelerating the drug discovery process in Alzheimer's disease. There are a number of different candidate drug treatments in development, but it's a real challenge to choose which ones to then progress and use in clinical trials. What stem cell systems allow us to do is to test which ones look like they work within the different Alzheimer's disease models, and that actually allows us to prioritize between the ones which look really effective and the ones which don't. And it's as important to succeed as it is to fail in a drug discovery early on in the process, so we don't waste time putting drugs into clinical trial which actually then aren't effective in patients.
One of the big challenges in discovering a drug to treat dementia is that we have to try to understand when the best approach or time to take the drug would be. And that really it requires us to understand what's going on within the brain as the disease develops. And so it's very important that we also carry out research in things such as imaging to help us identify patients who are at high risk of getting the disease.
We know that it's not really possible to diagnose dementia at the very early stages just from clinical signs and symptoms. And here we're using particularly brain imaging, but also other methods looking at changes in the blood to try and see which biomarkers are the best for diagnosing dementia at the early stages. So one very exciting development is that we're having 2 brand new scanners installed here in Cambridge. One is what's called a high field strength magnetic resonance imaging, or MRI machine, and that allows us to look at brain structure and function in much more detail than we're able to at the moment. And the second is a machine that combines MRI and another form of brain imaging, PET, and that will allow us in a single scan to get information both on the structure of the brain and also on the chemical composition and protein composition of the brain. So they're very exciting developments for us.
But drug discovery may not be the only answer to reduce the prevalence of disease. Despite predictions on the rates of increase of dementia, new studies show that improvements in our lifestyle are working. The number of new cases and overall numbers of dementia seem to be stabilising in some Western European countries. In the early 1990s, Professor Carol Brain and her team conducted a multi-site study across the UK to determine the prevalence of dementia in the over 65s. A recent repeat of this study has revealed some dramatic results.
What we found was that the amount of dementia, if you fix the age group, the amount of dementia that there is at any given age has gone down by more than 20%. And that means that something is happening within our populations and within our brains that may be protecting us. And to make predictions, we need to understand how whole populations have lived their life courses.
Professor Brain has used whole population studies to try to understand how early life, education, nutrition, exposure to alcohol and drugs, and exercise affect the prevalence of developing dementia. There seems to be one common link.
We know that educational level protects us from the expression of dementia during life. However, we don't know whether the education effect is because people who have education have to do more exercise, or it could be a whole range of different things. But we do know that having more education in early life, having a more intellectually stimulating job in midlife, and social engagement in later life, all of those things are associated with a lower risk of dementia.
The importance of a brain-healthy lifestyle is clear, and long-term studies are invaluable, but the burden of dementia continues to have an explosive impact on our global population. For the millions of people around the world whose lives have been directly affected by this devastating condition, it's vitally important that the search for new ways to treat and prevent dementia continues.
The vision that I would have is that we design our treatments and our approaches to support people with dementia and mental decline towards the end of life appropriately, so that we're not trying to, as it were, cure it with a single pill, but having a very smart approach to it, understanding that these things will need different approaches, but integrated.
It would be important for Andrew to know that there were more treatments available for people like Andrew, so that people can have a good quality of life as long as possible, and their families can have that member of their family, and their friends can have that person around for as long as possible.
And there's no reason why, given the level of understanding we now have about the biology of Alzheimer's disease, to my mind there is no reason why we should not be able to develop effective treatments for it in future.
We're very, very excited about being able to quite soon get these into patients and see if we can impact the progression. If you can stop progression and you can stop it in the same way that we've shown works in mouse models, it's incredibly exciting times.
What I think we should aim to do is buy individuals who are at risk of getting these diseases as many years of good life as possible. And so if we can delay the onset of the disease, we'll be accomplishing that. Indeed, if we can delay the onset of the disease beyond the normal lifespan, we've cured the disease.
I believe that Alzheimer's disease is eminently treatable. And by bringing together the expertise that exists in Cambridge from different disciplines is already leading to a new understanding of the origins of this disease, and that new understanding will generate new types of therapies, new drugs that will transform our view of this disease in the future.